So you should have heard a little voice say, a little automated voice saying recording in progress. This session is being recorded from now on. You will be able to watch back, and we'll send a link out so you can either watch it on YouTube or you can watch it on Zoom. So if you're joining from a part of the world where YouTube isn't accessible, you will be able to watch it on Zoom. Okay, now that we've started off recording, we're always really curious to know where our attendees are joining from. So if you're happy to, could you just in the chat let us know where you are in the world? I'm in southeast London, where I can see a little bit of blue sky out of my window. And somebody's on the Kent coast. Well, I hope you're enjoying a bit of sunshine down there. Somebody else is in West London. Somebody else is in Kent. Oh, two people from Kent. That's interesting. So from Colchester. Ah, right. Well, you see, this is why it's really helpful to know this. Belgium, Nottingham. So we've got a lot of people in Northern Europe and UK, which is, that I'm guessing the scheduling from this is quite good. Although somebody's just joined from Malaysia, so clearly the time zone thing is working for somebody way outside of Greenwich Mean Zone. Very warm welcome. The person joining us from Malaysia, you're definitely the winner of our competition for who is the furthest away from us right now. Thank you, that's very helpful. Right, what I'm going to do now is I'm going to launch a poll, and this poll is just to find out whether you have an offer to study with us. It's to find out whether you are, we just because we send out when we send out an invitation to this event, we send it out to people who have an offer to study with us. We also send it out to people who don't have an offer to study with us, and we're just really curious to know what sort of a mix that is. Whether some of the people who might potentially coming to do a degree with us have come along today. And I can see that from this, the majority of people don't have an offer, but some of you do. So it's great. So those of you who joined and have an offer, it's fantastic to have you here, and you'll be hearing some other things from us. Those of you who don't, I hope you apply to us. That'd be really good. Fantastic. So thank you for that. That's been really helpful. And I'm going to launch another poll now, which is going to be if you're interested. I'm guessing that most of the people here today are considering doing a degree. That's the kind of audience that we generally have. So what level of study would it be? Would you be more interested in doing like a BSc or an MSc? So it's just helpful for us to understand whether we're gauging the content of our presentations at the right level. And so far, the vast majority of people are considering doing a degree. Some people are considering doing an MSc. You will, there'll be aspects of this that will be at the high level, aspects that will be at the lower level. There's a real mix, but Steve will pitch it for a whole lot. And also, I just want to reiterate, if there's anything in this session that you don't understand, you think I don't understand that particular thing, if you were to do a degree with us, we would get you to that level. That's what we do. Our modules are very specifically designed to teach you the biology, the chemistry, whatever it is, to get you to the point where you're able to understand all of the concepts that we do in all of our taster lectures. So that's very helpful. Thank you for completing those polls. Very useful for us. And that little thing lets you know that I have my email on in the background. I'll give you my email address in the chat. So if you do have any problems during this session and you can't post in the chat or whatever, you can email me. I will get them during the session. So how's this session going to work? Once I've finished sort of talking through how this is going to work, I'm going to hand over to my colleague Steve. But if you have any problems, if you can't hear us or Zoom is acting up in any way, you can let us both know in the chat. If you're on a mobile device, you may not have noticed, but you've got a series of buttons at the bottom. You can choose to see us in gallery form or speaker form. It's up to you how you configure it. You've also got a chat function, and you also have a Q&A function. You can post your questions at any point. If there's anything you see in the slides which you want to ask about, or if you have any other questions, please put them in the Q&A box. You can ask them anonymously, but please put your questions in the Q&A box, not the chat, because the chat tends to sort of go like this, and we can't keep track of questions in the chat. So put them in the Q&A box. They can also be gathered up, and sometimes I can reply to them with a text message. That's easier. Once Steve's finished his talk, I'll come back on and I'll talk to you a little bit about the events that we've got coming up, and then we'll go on to our questions at the end. I will turn my camera off so as not to distract you, but I will be here in the background throughout the whole of this webinar. Now, over to you, Steve.
Hello, thanks, Kim. Hello, everyone. As we go in where we are, I'm out in Sussex today. because I would normally be in the department in London, but we don't have any trains today, so I'm out here. So I'll just share start the slides.
Yep, that's great.
Hopefully you're seeing that okay. So topic of today is going to be on vaccine manufacture, and what I want to try and explain to you is to why do you need engineers? So often when I'm giving these talks, I'll sort of do around the room ask of who's involved in vaccine manufacture, and often people will suggest, scientists, clinicians, all of the standard things that you might think, but often they don't really include engineers. So I want to explain today, give you an idea of why engineers are crucial to vaccine manufacture in particular, and biological products in general. So just to give you a little bit of background about myself, I'm not an engineer, but I've become very much an advocate for the role of engineering in biology. So I did a bachelor in microbiology and then a PhD in biophysics, and then spent quite a number of years traveling around the world, Germany, the US, and then back in Britain in Cambridge, Imperial College, MRC laboratories attached to UCL, studying basics of cell biology and particularly how viruses work and how they interact with cellular systems. I then moved out of academia into industry and was head of a biochemistry division of a company called iCure, which was developing based on what we call virus-like particles, which I'll explain a bit more about. And I've since returned to the Department of Biochemical Engineering. I was collaboration with the working in collaboration department during my time at IQ. We worked very closely together. We had numerous grants co-funded through the department and the company. Originally as a research fellow for quite some years, but now I've moved more recently as I've become a lecturer in the department and as actually the outreach and public engagement manager for something called the Vax Hub. Explain what that is in a moment. So I'm still very much attached, involved with research, spend most of the time now talking about what we do and how to explain it to the general public and the department. So what is Vax Hub? So what was originally called the Future Vaccine Manufacturing Hub, or Hub or Vax Hub, because of a mouthful, is an academic collaboration led by UCL Biochemical Engineering and the Jenner Institute at the University of Oxford. So I don't know if you've heard the Jenner Institute, but the Jenner Institute is one of the world-class leading institutes in vaccinology. They're behind the Oxford AstraZeneca COVID vaccine, that's the Oxford link in there. So they are very much fundamental hardcore vaccinologists. And our role at biochemical engineering is to provide the engineering input into take the vaccines into actual manufacture and use into the general public and for use through the world. So the aim are to do innovative research in developing, scaling up and manufacture of vaccines, particularly to low middle income countries where there's a great need for these vaccines. So as I mentioned, the work originally undertaken at the Vax Hub in 2018 and around to 2023, I'm very involved in the rapid development of the Oxford AstraZeneca vaccine, and for that we've been refunded and have now actually ready to split into brands of Vax Hub. One is called Vax Hub Sustainable, which is focused on sustainability of manufacture and supply. Vax Hub Global, which still has the original full development of manufacture for low middle income countries. So the hub vision is to make UK the global centre for integrated discovery through bioprocess production of next generation of vaccines. We do that through a number of ways: advanced high quality research technologies to bring new vaccines to ways to bring that new vaccines to market, to try and de-risk the bottleneck production existing production processes to make them more rapid and be able to respond more quickly to new solutions. To enhance production through responsible innovation and long-term partnership and upskilling, to be able to bring new people into the field with the skills that we need, because we have a constant need for new skills in this area, and also to promote better understanding of vaccines and the role of engineering in general. So that's now become my major focus, although I'm still very much involved with what's going on in the research areas within the hub. So I'm going to sort of split this lecture into a couple of parts, two parts. The first, I'm going to give you sort of an introduction to vaccines, what they are, and generally the types of vaccines and how they work, and then we're going to go into the second part, which is going to be more to do with the actual manufacturing processes, the challenges of vaccine manufacture. So what is a vaccine? We've all heard about vaccines throughout the world this last few years, but everyone, essentially, I'm sure, has some idea of what a vaccine is. But a vaccine is essentially, it's a way to trick your immune system into thinking that you've had a disease when you actually haven't had it. The idea being that our immune systems function to protect us from disease. They respond to infection, and hopefully they will overcome that infection. But there are always potential two outcomes to an infection. One is that your immune system can cope to handle it, and one, the pathogen wins, and you could have serious disease and ultimately death. We don't want that outcome. We don't want you to encounter a disease in order to get natural immunity. So vaccines are used to give your body the protection so that it knows how to handle a disease in future when you actually become infected by it without having to encounter that initial infection. A couple of terms that sometimes people get mixed up. So vaccination is the administration of the agent specific but safely and antigenic components that in vaccinated individuals can produce an immune response. So the actual vaccination is basically getting the injection. It's when someone comes along and injects you with the vaccine. Immunization, on the other hand, sorry, just to skip back there. An antigen is what we talk about, which is what gives you what your immune response reacts to. That can be a protein, a lipid, a carbohydrate, or a nucleic acid. So vaccination is administering that antigen. Immunization is when your immune system responds to that antigen, often produce the production of antibodies that can recognize something specific about that antigen, and then will be able to protect your body against subsequent infections against that antigen. Hopefully that's clear. If it's not, questions in the Q&A session. Feel free at any time to type stuff into the Q&A session, and I'll come to it at the end again to answer many questions as possible. So why are vaccines important? So vaccines are particularly important, obviously, to give us individual protection, but they're important on a communal level and a worldwide health level. Vaccines are essentially ways of preventing diseases. You get sick and then have to be cured or treated, but not getting sick in the first place. So if populations are generally healthier, don't get sick, that has huge implications on lots of things on generally how well the world lives. If populations are healthier, they're less likely to have poverty. If they have less likely to have poverty, they're less likely to be hungry. They're more likely to be even more educated. You'll probably have better gender equality and generally equality all the way around. I'm not going to go through all of these situations over here, but again, there are a number of sustainable development goals set back in 2015, and immunization is considered to be important in most of those, contributing to most of those sustainable development goals to promote better world health and better standards of living throughout the world for all of us. Vaccines generally take quite a long time to develop. Usually it takes 5 to 15 years, averaging around 10 years, sometimes longer. In the course of COVID, which we can discuss in the Q&A sessions if you like, it can be shortened. But that involves, because it involves many steps which are a part of the pathway. So we have a number of stages which are involved. You've got the initial R&D stage, which can take many years. It's involved in understanding the disease and developing things which could be potentially good antigens, which you can use to give a potentially good immune response. They will form the basis of the functional component of your vaccine. You'll then go into a preclinical stage where the safety doses are tested, usually in animal models. Sometimes if we can use cellular models, we will do, but the immune system is quite a complex thing. And there are few pure cellular models that can be used. And often, to be quite honest, we have to use some sort of animal model. Again, this can take several years to work out which are the potentially good vaccine candidates, which will be then taken through into human clinical trials. The first of these is what we call phase 1, which is assessing only essentially safety. You're assessing on a relatively small number of people, you're testing out different doses, and you're measuring, are there any adverse side effects? You're not really assessing at this stage, are you getting any good immune response, but you're generally testing just safety of side effects. You can move from, if that phase is passed, you'll move into phase 2, again, where you'll start to increase your population size, and you'll again looking for any potential side effects, and starting now to look at the effects of different dose sizes and starting to measure the immune responses. Phase 3, you go again to a larger population, can take years to decades, depending on the type of disease which you're studying. Again, always you're noting this, that safety is first. Always we're testing if there are any potential side effects, because we would remove anything that was giving us a bad safety profile or not acceptable safety profile. We're also then testing efficacy in our target population. Are we getting the immune response we get? And importantly, are we getting protection to the disease? If all of those turns out to be positive, you'll get licensing and approval. You'll submit your data for approval at regular. See, they will approve it. But even then, you've got to remember that an approved vaccine is not necessarily approved forever. Monitoring of all vaccines continues after licensure and rollout for general widespread use. And that is also always measuring effectiveness and, importantly, safety. If we're getting any unanticipated adverse effects, then we can change the licensure either to restrict that vaccine so that particular groups don't get it, or it can be completely removed from the market completely. And that has happened, that does happen with vaccines. So what sort of percent of vaccines actually enter clinical trials and successfully make it through to phase 3? It's actually quite small. It's less than 5% of vaccines will make it all the way through. So vaccine development is lengthy and only has about a 5%, less than 5% success rate. So we need to be getting lots of vaccines going in at the R&D stage to get enough vaccines coming out at the approval and licensure stage, and to go into general world use. Now, as I said at the beginning, you might sort of think about, who thinks about who's involved in vaccine manufacture. Often people say, researchers, developments, biochemists, medics, and then what we're going to talk about particularly more is the role of biochemical engineering and engineers, statisticians, obviously, for the data analysis, clinicians, doctors, always important. But there's a huge array of people who are actually involved in these things. People who need to work in the regulatory agencies, the policymakers. Like any marketed product, you've got to have patent attorneys, you've got to have funding, and you've also almost going to have move this out on scale. You're going to have mass production. You're going to have a large pharma companies. So just put this up because if you're sort of interested in the vaccines and vaccine manufacture in particular, don't think, well, I don't want to be a researcher, or I don't want to be a microbiologist, I don't want to be an immunologist, I don't want to be a clinician, but I'd still like to be involved. There are lots of areas where people potentially have very significant roles in vaccine development and production. So that sort of gives you a little bit of background on what vaccines are. Today is going to be, relatively superficial. We're not going to go into great detail. As Kim said, if you are studying this at degree level or beyond, obviously you are studying in much higher detail, but today we only have 30, 40 minutes or so. So we can't give you a whole course in that. So it's going to be very sort of basic level. So different vaccines, there are different types of vaccines. When I say a vaccine, there are lots of different types of vaccines. A vaccine they all have the same function: to stimulate your immune system to protect you against future exposure to that disease. But there are lots of different technologies which we can use, which may be appropriate depending on the type of disease, the pathogen, whether it's a bacteria or a virus, for example. The type of technology, it can affect the safety profile, and it can affect its accessibility and affordability, which are very important if you're going to roll out to worldwide use. So current vaccine technologies, we have a whole array of different ones. Often people, and I know at our sort of GCSE level within the UK, our definition of vaccines are that they are live or inactivated viruses. Those were some of the earliest of vaccines. So the earliest vaccine was against smallpox, the work of Jenner. That was essentially an attenuated vaccine. Some of the earliest of vaccines are essentially alive or inactivated viruses. An attenuated virus is a virus which has been, or a pathogen which has been weakened, so it no longer causes a significant disease, but your immune system will still see it as looking very like the dangerous pathogen, so it will give you a response to it. An inactivated vaccine, for example, can be the actual pathogen which has been killed, inactivated, so that it's not dangerous in any way, but will still give you a very good immune response. Some of our earliest and most effective vaccines are based on these type of technologies. They're very good at giving immune responses, but from a large-scale manufacturing process point of view, despite the fact that they are still used, they have one big disadvantage, being that in both cases, particularly in the inactivated vaccine case, before it's inactivated, you are producing large amounts of potentially deadly pathogen. An attenuated, a weakened form of pathogen, if it's been weakened genetically or by some sort of mutation, there is a possibility of what we call a revertance event, whereby that weakened form can regain function that we've weakened and become also more dangerous. So in modern vaccinology and vaccine manufacture, we like now to try and move away from these situations and to look at more safe profiles. So the first of those is what we call a subunit vaccine, whereby you, instead of manufacturing the whole pathogen, you're just manufacturing individual antigens, usually individual proteins, which will give you an immune response. Or maybe if you're looking at a bacteria, you might be producing polysaccharides to give you immune response. These are very safe, but because they don't have the complexity of structure that you see with a whole pathogen, your immune system often doesn't see them as well and give a stronger response as it would to a whole pathogen. So we try to have ways around that. One is which we call a virus-like particle, which is essentially a virus taken, the genetic material has been deleted so that you have the structure, your immune system sees the structure, but the pathogen no longer has its genetic material, so cannot replicate. Then we have viral, what we call vectors delivery systems, which can be viral vector, DNA vaccines, or mRNA vaccines. The difference between those and the others are essentially that you're delivering a message for the antigen that you want to use, and that you're generating that genetic message, introducing that into the cells of your body in such a way that you will get a short time length expression of that pathogen, of that antigen rather, to give your immune system a booster and an exposure briefly to that antigen, again, to generate a protective immune response. Each of these types of technologies have individual manufacturing challenges. I'll just give you some examples, and all of them are essentially used. So live attenuated vaccines are still very much used for measles, MMR vaccines, chickenpox. Inactivated vaccines, still very much used for polio and influenza. Subunit vaccines, we have many different types. We have polysaccharides, which often, and conjugate vaccines, which use mainly against bacteria. We have protein-based vaccines, which can be used against virus, or used against, often against viruses. Viral vectors, originally been used against Ebola and COVID. mRNA so far has only been used against COVID, but new ones are in development on the success of that. VLPs, we have some very effective vaccines against, particularly against HPV, hepatitis B, and some new vaccines which have recently come out, which are going to hopefully give a significant effect against malaria. We have no human vaccines which are based purely on DNA technologies, but there's lots of work, and also work within the Vax Hub that maybe hopefully will bring those technologies. Again, just to give you a point when I say about a vaccine. So a vaccine, and not all vaccines are the same. And a vaccine is not just the antigen. A vaccine is the whole component. You have to have if you're using a subunit, for example, as I talked about, they are effective immunogenetically. So you have to give things which we call adjuvants, which stimulate the immune system. You may have preservatives in there, which would need to use. Have we got an issue? Because I can see a hand raised somewhere. Okay. Hopefully you're still hearing me. If Kim's not, he'll tell me. You can have preservatives in there. You can have stabilizers. Anything, because you be able to store vaccines and transport them, so you have buffers and so forth. So a vaccine itself has the antigen active component. It can have lots of other things. All of these have to be part of the manufacturing process. And these are part of what we call the final formulation process. So what characteristics do we need to have an effective vaccine? Obviously, it needs to be effective in preventing or reducing the severity of the disease, else it has no biological function. We want that protection to be durable, so we want it to give long-term protection. Our ideal vaccine is something that you have once and you're protected for life. There are vaccines which will do that. Measles, for example, is a measles vaccine is very good, giving lifelong protection. You want the active immunity with a minimum number of doses. People don't like to keep being immunized, having more and more doses. It's inconvenient, it's uncomfortable, and it can make just the process of getting vaccinated difficult. The more you want the least number of doses that you can get away with. You want to provide the maximum protection against a number of pathogens. For example, broader pathogens. Influenza is the common one. We have lots of different types of influenza viruses. We want vaccines that will give us the broadest protection against encountering not just one type of that pathogen, but as many of them as we can do. We obviously don't want severe or side effects. has to be stable, has to be able to be stored and transported to where it needs to be used. Yeah, in Sussex, where I am, my local clinic has freezers, but if you're out in other parts of the world, you may not have access to that, and simply getting material to where it needs to be used, and it needs to be able to be stable. It has to be available for general use through mass production, so we have to be able to manufacture enough of it that we need when we need it. It has to be affordable of populations at risk. So particularly if populations are low income, they need to be able to afford these vaccines. And most importantly, can have an effective vaccine, but it'll have little practical use if we can't manufacture it at enough of it at the scale that we need and at a price that people can afford. So that's where the role of what I'm now going to concentrate on, biochemical engineering, comes in. How are vaccines produced? What's the secret behind making millions of doses? That's large-scale part of biochemical engineering. So that biochemical engineering essentially covers the whole process all the way through. Biochemical engineers, as we say here, apply the principles of biology, chemistry, engineering to produce products. Not just related to vaccines, biopharmaceuticals, fuels, biopolymers, industrial enzymes, alternative meat products. I think there's a talk possibly tomorrow or later this week on just that. And it covers the whole area of what we call synthetic biology, which is working with molecular biologists, biochemists, biophysicists. Remember, my background is very much in molecular biophysics, but I've become much more involved in the process side, as my involvement, particularly in industry, was very much for how do we take our discovery from the bench, this VLP-based vaccine, into manufacture so that we can make use of it at a worldwide level. So you've got lots of interface with biomedical scientists and clinicians at the end. So biochemical engineering covers all of these interactions. The questions that we're going to have to answer is, how are you going to make what is this wonderful vaccine that you've got? What's the right technology to use? And one of the key questions is, how much can we make? How much do we need to make? What is our target population that we need to protect? How can we ensure that the product is going to be safe and make the regulatory requirements? And crucially, how much is it going to cost? How can we make it affordable? So a lot of our engineering challenges are in the costs to make healthcare accessible. We need vaccines and therapeutics to tackle new diseases in specific parts of the world. And we need to make them sustainable chemicals, and we produce them sustainably, not to be reliant on petrochemicals, for example. And also often the technologies, I'm just not going to talk about gene therapy today, but the technologies that we're involved in, things such as gene therapy and targeted healthcare, are very much overlapping with the technologies that we use to produce vaccines. Not identical, but very much overlapping. So if you're studying one, it's very easy to study and move into another field. So biochemical engineering covers the whole process of production, and we tend to divide that up into two phases. We talk about upstream processing, which is essentially going from the first steps of bioprocessing. Remember, these are biological products, so you're normally using a cell system, so you've got cell isolation and cultivation through to development of cell lines, final harvesting. So upstream is basically when you're dealing with something that's alive, that's making the product you want, the vaccine, the product you want. You start off on the milliliter scale and can go up into the hundreds of thousands of liter scale. Second half of the process, what we call downstream processing, which is when you've got your cellular system which has made the product you want, is how you then separate that product that you want away from all of this. And for that, we use isolation and purification technologies, which are important in terms of how we're going to separate what we want from what we don't want. We have a whole range of technologies which are involved, which we can do different stages of that. Each of these could be a separate talk on themselves, but also at the end of it, we shouldn't forget about the quality control and the formulation and package we've got to package and port our product to where we need it to be. So I'm just going to give you a little quick overview of the top technologies involved in firstly, the upstream side of things. So remember that upstream side of things is the first step of bioprocessing from the earlier producing cells, which are going to give us the product that we want. It's going to be expressing our VLP, our subunit, our virus, or whatever. And this involves growing cells to give us the product biomass that we need, the vaccine yield that we need, and importantly, the cost. One of the big challenges is to improve overall productivity to decrease costs. And that involves obviously going from relatively small scale cultures up to cultures which could go up to thousands of liters, tens of thousands of liters. To give you an idea, within the department that we go to, we can operate. We can go all the way from lab scale, a few hundred mils, all the way up to 100 liters of culture to optimize our growth conditions. Essentially, we'll be using two types of expression systems. We'll be using microbial cell culture, or we'll be using mammalian cell cultures. Each have their advantages and disadvantages. The microbial are fast to produce, fast to grow. They have simple process needs. They have and they're cheap to produce. But if you're producing a complex antigen or a complex product, you may not be able to produce that complexity to get the correct structure, which is what you need for your antigen to stimulate the immune system in a microbial system. So you may have to switch to a mammalian cell system, but then you'll have greater complexity. They're much more sensitive to grow, they're slower to grow, and they're much more expensive to produce. So always we're looking for the correct balance between what type of expression system to give us product quality at the price and at the scale and price that we want. Downstream processing is now then part of how we isolate and purify our vaccine away from what we want, which is essentially stepped up into a number of steps. What we call primary recovery is where we isolate the cells. We can engineer the system in two ways. We can try and what we call the vaccine will be secreted out of the cells. So it will be produced in the cells and then secreted out into the media, which makes it much easier to isolate. But getting complex structures, we may not be able to do that, and they will be contained within the cells, and then we have to be able to break open those cells and isolate what we want from all the rest of the cellular components. So either way, we will have a number of purification steps, and then finally we will have that formulation adding to our pure product, the adjuvants and excipients to give us our vaccine product, which can then be shipped for use. The types of technologies that we'll use for downstream processing include centrifugation. Again, you're probably familiar with this at a laboratory scale. On the example on the left, it gives you the laboratory scale, what common use, an example of the sort of manufacturing large scale of type of equipment that we have within the department over on the right. Filtration is another type of technology which is often used to initial stages of purification. After following centrifugation, filtration to remove smaller then finally, we'll use the often different combinations of chromatography, a method of separating molecules by their physical chemical properties, such as charge, hydrophobicity, size, or some sort of specific fit that they'll have for some sort of component which can be engineered into the system. Now, one of our biggest challenges is scale. It's how you translate manufacturing to bench scale laboratory work. Obviously, we want to be able to do experiments at bench scale to optimize our conditions. We want to do lots of experiments to work out our best upstream and downstream conditions at small scale. We don't want to be doing experiments on 100 liters or 1,000 liters. That's wasteful, it's time consuming, and you can't generate. So we want to be able to generate lots of data at small scale, which is going to be easily transferred, easily scaled up to full manufacturing scale. And that is harder than you may anticipate, you may think about. Because imagine I don't know how the example I like to give for those of us who are fans of the Ian Fleming books and the old James Bond movies. The idea of martinis being shaken, not stirred, because the taste is different. That is true, actually, and it is also very much true of biological products. Imagine you have to mix something. If you've got a liter or so, you can mix it by simply shaking it. If you have to mix 1,000 liters or 10,000 liters, you will not be able to shake it. You will have to have some sort of impeller system to mix it. And the way that change from that type of mixing from shaking to impeller mixing can give you differences in your product. So what we need to be able to do is, instead of what we call scaling up and then finding out that we hit these technology bottlenecks where we can't go from a mixing technology because we can't shake anymore to a impeller, and then all the data you have on shaken is not as relevant as you need to scale up, is to draw this paradigm of what we call ultra scale down, which is to break the paradigm of development and testing at lab scale. And the philosophy is to assume your product is going to be good, and then think about how much do I need and at what price and what technologies are going to be appropriate. And how do I not scale that product up, but scale down those technologies to be able to operate not on 1,000 liters, but on 1,000 milliliters, so that I can generate lots of data. So a lot of our scale up technology experiments are actually on generating technologies which can allow us to scale down, so that we can do microscale and DOE to give us lots of map of large knowledge space, which we can then translate easily to full-scale manufacture. And a large influence within Vax Hub and within the department is the generation of these technologies, which are now can be successfully applied to a whole array of stages, unit operations within the whole manufacturing process. What we then try to do is to choose a much more holistic approach, again, to use data-driven approaches, lots of data-driven approaches and data modeling to be able to accelerate the whole developmental production scale. So what your molecular design can impact your engineering. That can maybe impact your stability and formulation and feedback to your cost and your process development. So what we try and do also is to model these processes so that we can work out, again, the most efficient way that's going to give us the product that we want, the effectiveness that we want, the safety profile that we want, on the scale that we need, and a cost that people can afford. And engineers play an essential role in bringing life-saving vaccines to widespread societal use using these principles. So to wrap up there for today, if you're interested to learn more, and if you are enrolled on one of our courses to be one of the people that have an offer from in the final years, we do offer a 40-hour full vaccine module on vaccine bioprocessing, which is taught by Dr. Stephanie Frank and myself. If you're not involved in our course, but you would like to learn more, there is actually a shorter version, which is a free online course, which is available from the UCL courses platform, called Towards Improved Tools and Technologies for Vaccine Manufacture. That's completely free. You can sign up to that. If you go to UCL short courses and just search for vaccines, you'll be able to find it there. So thank you for your attention. I'll wrap up there, which will give us a few minutes for questions, hopefully, and go from there. Thank you.
Great. So thank you very much, Steve. I see we've got some questions that have come through, some really, really great ones already so far. Before I go into what I want to talk about next, somebody's asked, what careers do biochemical engineers typically go into? We'll go into that in a really deep dive at the UCL undergraduate open day, and I'll stick a link to that in the chat, because you can sign up. Myself and Dr. Jack Jeffries will be online one morning session, one evening session, so that you can come along. We'll talk to you about careers, modules, the two different types of degrees we do, and you can find out way more about it then. And we can talk to you about all the different career opportunities that some of our graduates have gone on to, whether it's working in the lab-grown food industry or pharmaceuticals, or really wide range of stuff.
Mhm.
We can go into way more detail on that.
Yeah,
I'll
and
put it.
just like to add on that, Kim, remember that link back to that slide I showed, the fact there are all of these arrays of people that were involved in, just using vaccine manufacture as an example. I think we probably have graduates who have gone into almost all of those fields. So again, they can give you some example of what we do.
Great.
Hmm.
What I'm going to do now is we're going to hand over to Steve for the questions. And once as Steve goes through those, I'll pop a couple of links in the chat so that you can, if you want to register for the undergraduate open day, come along, see me and Jack. And the other thing that's happening on the 28th and 29th of June, we have an in-person open day. And for that, you'll actually be able to visit our pilot plant, which is an incredible opportunity, incredible facility. There's no other university in the UK that teaches biochemical engineering from a scaled-down pilot plant. Meet the team, have a look at the campus, and that's over 2 days at the end of June. So if you can come to London, it's well worth it. It's a really good opportunity to find out all about what we do here in person. But Steve, I'm going to hand over to you.
Okay, so I'm just looking through the Q&A session here. So I'm going to work my way through. There's not too many. Hopefully, we'll have time to get through most of them. So the first one I'm looking at here is: do you feel that mRNA vaccines are the way of the future? Potential benefits on our challenges for mRNA vaccines compared to traditional vaccines. So the first thing I'm going to say on that is that mRNA vaccines have really came to the fore in response to COVID. They were a technology that have been in development for a couple of decades. The thing that was holding them back was very much the stability issues. They were not particularly they were effective, but not particularly stable, which meant that they were hard to administer. That was brought a long way forward with the use of lipid nanoparticle technologies to encapsulate them, which has vastly increased their stabilities. Although at the moment, the ones which are on the market still do require very low temperature cold storage, which again makes them hard to administer to parts of the world, to roll out to lots of the world which simply don't have those facilities. mRNA vaccines certainly have a future. I think at the moment there's a lot of work going on into mRNA vaccines. They have manufacturing advantages, that they are potentially simpler to manufacture because they are essentially synthetic products. They are not, they're not part of a traditional bioprocessing in the sense that they're not produced in a cellular system. It is much more like synthetic chemistry. So they're much easier to produce in that respect, although they are still expensive because some of the chemicals involved are expensive and hard to get. They will certainly have a future. There's a lot we still need to know about them. The only example we have at the moment is COVID, where they have been very instrumental in helping to tackle that. But one of the things that we are finding there is that they're not giving, but I've talked about durability of response. They're not particularly giving very long, durable responses, which is again part of this idea that we have people are having to have relatively frequent boosters, which is not our ideal vaccine. Our ideal vaccine, again, is something that you would have once and then be protected for life. Now, that could be a feature of the particular COVID antigens we were using, or it could still they're very much open whether it is a feature of the way that mRNA vaccines stimulate the immune system, which they are not in themselves going to be good for generating highly durable responses, but will remain to be very good at rapid response to outbreaks. So I think there is definitely a future for mRNA, but I think there's a lot more we need to know about it. Can antibiotics increase? Can antibiotic resistance increase from vaccines? I would actually say the converse is true. So a vaccine will generally not be affecting antibiotic. It will not make your pathogen more resistant to antibiotics. What we're tending to think now is that vaccines have a very potentially important role in combating antibiotic resistance, because antibiotic resistance is essentially potentiated by the need to use antibiotics. So if we decrease the need to use an antibiotic by the fact that you get less people sick because they've been vaccinated and are protected, then you will decrease the spread and the speed of the spread of antibiotic resistance. Again, there's a whole half of one of our lectures is on this topic and it's explaining why that's why. But the concept there is that vaccines are actually going to be very, very important in helping us to combat the spread of antibiotic resistance. Someone here is asking what types of polysaccharide are found in polysaccharide vaccines. So the polysaccharide, a lot of bacteria essentially have polysaccharide on their surface. Your immune system, that's the thing that's the structure that your immune system sees. It's not protein, it's polysaccharide. Those polysaccharides can be very varied in structure, and therefore we need to try and produce antibodies to produce vaccines to produce antibodies to polysaccharides. Unfortunately, polysaccharides, the way they stimulate the immune system, does not give us very long-term protection. Again, so what we try then to try and get around that is what we use, what we call conjugate vaccines, which are polysaccharides from a pathogen conjugated, physically coupled to a protein, which we know is going to stimulate the immune response and produce vaccines that way. Some of the most important vaccines which are out there are based on protecting against bacterial infections are based on those types of technologies. The manufacturing challenge there is that you have essentially a threefold component system. You have to produce the polysaccharide, you have to produce the protein, and then you have the chemistry of coupling the two together to give you your final product. So you end up with a very, very complicated and time-consuming and expensive production methodologies for those types of things. If I can scroll down. What topic did I cover in my PhD? What part of biophysics? Okay, my PhD was on defects in structural proteins which lead to hemolytic anemia. So it's very much on protein structure and protein physical chemistry, and that's quite a long time ago. But so I've moved a long way from that now. But thanks for asking. Another question on antibiotic resistance, which I hopefully have sort of covered. Do you think stem cells are useful in testing vaccines in the lab? We're constantly trying to move away from using animal models, and stem cells may be useful. The problem is that our immune systems are such complex things and that involve lots of interactions that producing really good non-animal models for the immune system is still difficult, but there's lots of work going into that way. Do you believe that social media and online activity is increasing fear-mongering and causing more people to refuse vaccinations in the modern day? Yeah, the short answer is that some of it is, but hopefully what we're saying with our sort of trying public information campaigns and lots of more public information campaigns, hopefully we could try and counter some of that. Often you find is that when you talk about refusing vaccinations, we talk about vaccine hesitancy. I prefer to talk about vaccine hesitancy rather than anti-vax. There are some people who are simply against the idea of vaccinations, and then for that they may have very valid personal reasons for that. But often what you find is that when you really drill down into this, it's the fact that people are vaccine hesitant because they don't feel they have enough information, enough understanding of how it works and what the advantages and disadvantages are, what the risks are, or the risk benefit, what's the risk of the side effects, exposed to the risk of catching the disease and the risks from catching the disease. And then there are simple things, but very important things that gets in the way of talking about, if you have a vaccine, which means you have to go a long way to a clinic because the vaccine is hard to transport. So you have to go, for me, Sussex, I just go down, I can walk down to my local vaccination clinic round the corner. There are lots of parts of the world people may have to travel for days to get to the vaccination clinics. Again, if you have one vaccine, you need one dose, protected for life, wonderful. If you have to keep going back and back and back again, the chance of getting overall protection become much higher. So the better we can engineer our vaccines to be more accessible, the better coverage we will get. Okay. So I don't think I've managed to cover everything. But
I'm just going to nip in to say somebody's asked a question whether there be online alternatives to the open day. Yep. So the first link that I've put in the chat is the 23rd and 24th of April. That's online. Will be online. There's actually, what we've done this time to try and make it just a bit more effective. My colleague Jack Jeffries has pre-recorded an introduction to our degree programs. I mean, that covers a huge amount of stuff. The content of the program is what people go on to, and then we'll be online for half an hour, so you can ask us any questions, anything, if you want us to go into more information, if you've got any questions, then we're there to talk about that in detail. But yes, that's entirely online because, as we found out today, we've got people joining from all over the world. It's not viable for everyone to get to London. The one in London it takes so much time to organise it because we have tours of the labs. We have students who are coming and talking as well. It's really difficult. It takes a lot of our time, which is why we only do it once a year. So, we really do want to spend as much time as possible either teaching or researching. So, that we try and get the balance right. But yeah, there's two online ones. If you click the link, 23rd and 24th of April for online, and then 28th and 29th of June for virtual. So we've got 3 minutes left. Steve, I'm going to leave it to you to pick either two quick questions or one long question to finish up on.
But I just saw one quick. Somebody was asking about do we have a program for education in secondary schools or colleges? We are rolling that out. So at the moment, we have only rolled out within the London area and Stevenage. But the Vax Hub we are hoping to roll that out but much more. If you're interested, if you're in the London area or Stevenage and you're in a college or school that wants to get involved in that, then please do. The easiest thing is either reach out to me or just generally to the Vax Hub website. Where if you go to the departmental website, you will find the Vax Hub website there, and you can just reach out to us. There's a couple more. What's it? Somebody was asking about quite a number of antibiotic resistance. Hopefully, I've covered that.
Yep.
Gene therapy. What about gene therapy? Gene therapy. I'm sure we probably have. I'm sure we have a whole lecture on gene therapy.
Yeah, that was the one that was on Friday. So what I'll do is at the end of this series, I'll send you a link and you can see the recording for that one. So Pedro covered a lot of stuff on stem cell therapies.
I would say there is a big overlap between technologies used for particularly sort of the vaccine, the viral vector delivery systems and the gene therapy systems. There is one crucial difference between using them for vaccines and using them for gene therapy. In a vaccine, you want a short-term expression of your product because you don't want to be constantly stimulating the immune system. So we want to design if we're using a viral vector for vaccine use, we're designing that not to integrate into your genome, but to give you a short term of expression so that your immune system sees that antigen for a short time, gets primed to see in the future. Gene therapy, obviously, you need to either to replace the defective gene, to integrate the new gene into the genome, or to be having your product being produced constantly so that you are constantly treated for that. So the technologies are similar, but not quite the same. They're used somewhat differently. Okay, I think that's hope that you can
Yeah,
always reach out to us with anything else. I'm sure we'll
yeah,
do that.
I'm really sorry we didn't get to go through all your questions. We would have loved to. It's just time constraints, but I mean they've been brilliant. Really appreciate your engaging with us. It makes it so much fun for us to do that when you ask these questions. Yeah, thank you for joining us. It's been great to have you with us. I hope this is the start of a fantastic week for you, and I hope to see some more of you at our other sessions. And I would be
Yeah.
great if we could see you in person at some point at UCL. But until then, thank you very much. Have a great week, and thank you,
Thank
Steve.
you. Okay. Thank you.
Bye bye.
Bye.